All right, it's my pleasure to introduce Brady Yokom. He is a graduate student in the Aquatic Animal Health Lab at Michigan State University, advised by Dr. Tom Locke. His research interests involve understanding the ecology of fish pathogenic bacteria, including Flavobacterium psychrophilum, the etiologic agent of bacterial cold water disease, a problematic pathogen in salmon aquaculture worldwide. Brady's also interested in the isolation of novel bacteriophages, which may be used for treatment and/or prevention of bacterial cold water disease. Looking forward to your presentation.
All right. Thank you, Dr. Phelps, for that wonderful introduction and to all the organizers for putting on this seminar series. So I'm going to share first some findings of an undergraduate research project that I undertook involving lake whitefish and Renibacterium salmoninarum. And then at the very end, I have just a couple slides that's sort of prospective in terms of what I'll be doing for my graduate research. But before I jump into the lake whitefish material, I'd like to take a second and thank all my co-authors for their help, because without them, none of this work would have been possible. So a little bit about me for those of you who don't know who I am. I'm a Michigan State alumni, and I've worked in the Aquatic Animal Health Lab for just under 3 years. Like I mentioned, this work that I'll be sharing today was part of an independent study with Dr. Locke this past summer, and that led to me transitioning to a graduate student role in his lab, which is what I'm doing now. So this is the lake whitefish. For those of you that aren't familiar with it, it's a keystone species in the Great Lakes, and it's critically important in terms of both the ecology, the economy, as well as the culture, not only for Michigan, but the entire Great Lakes region as a whole. Unfortunately, though, lake whitefish recruitment has been on the decline for a number of years and continues to decline today. So I'm sure that most of us are already aware of what recruitment is, but just so that we're all on the same page, it's the number of juvenile fish surviving to adulthood, which is indicated at this transitional phase here. As an unfortunate consequence of reduced juvenile recruitment, there's also been observed declines in adult lake whitefish abundance in the Great Lakes. There are several hypothesized factors that might be contributing to these declines, several of which are the invasion of dreisseniid mussels, which has led to a decline in diperized zooplankton, which is a preferred prey item of the lake whitefish, as well as climate change and all that that entails for the Great Lakes ecosystem. One potential factor that we don't know much about at all, though, is disease. And I'd like to point out that there are several diseases in the Great Lakes that have potentially detrimental effects at a population level for fish. So certainly not out of the realm of possibility that something similar might be happening with lake whitefish populations. Previous surveillance efforts have been undertaken and identified a number of pathogens, including Renibacterium salmoninarum, which is a gram-positive diplobacillus bacterium, responsible again for bacterial kidney disease in salmonid fish. But to date, all we really know about BKD in lake whitefish is that it's been detected previously in wild adults and was associated with damage to the kidneys. We know a little bit more about BKD and other salmonids, namely that it's been associated with substantial wild fish kills previously in the Great Lakes, and it's also been problematic in state hatchery systems recurrently over the years. We also know that the bacterium can effectively vertically transmit from infected parents to naive offspring, thereby perpetuating infection through generations. This hasn't been proven conclusively in lake whitefish, but the fact that it occurs in closely related salmonids means it's certainly possible that the same phenomenon occurs in lake whitefish. So up to this point, we've established that Renibacterium has been detected in wild adults previously, and that it's been problematic in other salmonid species in the wild previously. But again, beyond this, we really don't know much about bacterial kidney disease in lake whitefish. So along those lines, I hypothesized that Renibacterium would in fact be able to cause disease and subsequent mortality in lake whitefish. And to address this hypothesis, I experimentally exposed lake whitefish to the pathogen and then tracked disease progression and mortality. Now, I'm sure that most of us are probably already aware of Koch's postulates, but just to make sure that we're all on the same page again, we need to fulfill Koch's postulates to conclusively link a pathogen as causing a disease in a specific organism. So those consist of 4 criteria, the first of which is the pathogen needs to be present in sick individuals and absent in healthy individuals. You need to be able to recover that pathogen, grow it in the lab, re-expose it to naive individuals and observe them becoming diseased, then re-recover that same pathogen and confirm its identity. Now, to fulfill Koch's postulates, of course, we need to determine an exposure out to naive individuals. And in our world, this usually comes down to either immersion or injection. Now, for our initial pilot experiment, we weren't sure which, if either of these routes would be efficacious. So we decided to ultimately choose both for our pilot and see, again, which one was most effective at causing disease in lake whitefish. Now, to give you a sense of what that actually looked like, on the left are the actual experimental tanks in our laboratory. And then on the right is a diagram showing you the treatment that each group of fish received. And I'd also like to point out at this point that the bacterial isolate that we chose for these challenge experiments was recovered from a naturally infected Lake Huron spawning Chinook salmon in the Great Lakes. Now, in terms of pilot results, we noted apparent disease developing in all of our Renibacterium exposed fish. This included a variety of symptoms, including ulceration of injection sites, varying degrees of dermal hemorrhage, and multiple abnormalities in the kidney. And importantly, we were able to detect the bacterium in all of our exposed fish and in none of our negative control fish. Now, when it came time to performing a full-scale experiment, we were again faced with this decision between immersion or injection because both routes seemed viable, but we ultimately chose immersion because it more closely mimics a natural route of infection. Now, to give you a sense of what the full experimental setup looked like, here it is in the laboratory. So it consisted of 18 tanks divided into 6 triplicate groups, and the numbers along the top indicate the bacterial concentration that each group was exposed to, or the negative control group, which was all the way to the left. Now, externally, disease signs began developing roughly 1 month post-exposure and included things such as varying degrees of scale loss, which in some cases was more severe and also accompanied by hemorrhage. Severe ecchymotic hemorrhage was also noted in a number of fish. Internally, we noted in a subset of fish cream-colored nodules in the kidney that closely resemble the granulomatous type lesions that are typically associated with bacterial kidney disease. Oftentimes, or sometimes, excuse me, they had a more multifocal aspect to them, whereas other times they had a more coalescing nature. And it's pretty evident that lesions as severe as these would undoubtedly be causing extensive damage to the kidney organ and hindering its ability to function properly. Something that you might have already picked up on, but I want to draw your attention to in case you haven't. The kidney lesions that I was observing in my experimentally infected fish are very similar to those that were observed previously in wild adult naturally infected lake whitefish. And now I'd like to walk through the mortality data for this experiment. So on the x-axis is days post-exposure, and on our y-axis is our cumulative percent mortality. So starting with our negative control group, we did have some fish die in this group, but as you'll see in a second, we never detected Renibacterium in any of these fish. Next, our highest challenge dose, which reached 100% mortality at approximately 75 days post-exposure. And then our remaining challenge groups, which were all clustered around 30 to 40%, depending on the bacterial concentration that they're exposed to. Now, if you recall Koch's postulates, the final criteria of that was re-recovery of the pathogen and confirmation of its identity. So to do this, kidney tissues were streaked onto modified kidney disease medium from all experimental fish. Representative bacterial isolates that had morphology consistent with Renibacterium were then subjected to DNA extraction and Renibacterium-specific quantitative PCR to either confirm or refute the identity of those bacteria. As an independent, or a culture-independent confirmation technique, we also used the ELISA assay, or enzyme-linked immunosorbent assay, targeting specifically the P57 major soluble antigen of Renibacterium. And I know that most of us are probably familiar with this assay, but just as a reminder, the darker wells indicate a greater concentration of antigen in a given sample. So it gives you some sense of how infected the fish might have been. Now, breaking down detection by treatment group. So beginning with our negative and our lowest exposure concentration. So no detection of the bacterium via culture or via ELISA. But from that point, as we increase the bacterial exposure concentration, a noted gradient of infection was observed. So the greater bacterial concentration fish were exposed to, the higher percentage of positive via culture and ELISA we observed in those fish, as well as an increase in the number of high and medium ELISA positives for those infected fish. So in conclusion for these efforts, so it certainly seems that Renibacterium is in fact virulent to lake whitefish, at least at a sufficient concentration, and it may indeed be contributing to juvenile lake whitefish mortality, which could, at least in some capacity, be contributing to the observed decline in lake whitefish recruitment. Now, acknowledgements for these efforts. So I'd like to thank the Great Lakes Fishery Trust for providing the funding, for these efforts, the Little Traverse Bay Band of Odawa Indians for providing the fish used in these experiments, my colleagues in the Aquatic Animal Health Lab for all of their help to make these efforts possible, and then, of course, my major advisor, Dr. Thomas Locke. And now quickly, I'd just like to introduce everybody to what I'll be doing as a graduate student. So like Dr. Phelps mentioned at the outset, I'm going to be focusing on the ecology of Flavobacterium psychrophyllum. We know more about this in vitro, and its strides are being made every day to better understand this aspect of the bacterium, but much is still to be learned about the bacterium's ecology in aquaculture settings. So I'm going to be trying to understand the interaction between the pathogen and the environment as it relates to disease in the fish host. To do this, I'm going to be performing molecular surveillance using quantitative PCR by sampling water in aquaculture facilities, filtering that water to concentrate the bacteria, and then again, using quantitative PCR to quantify how many cells are present in that water. The overall goal of this data is really to be better able to anticipate when and where disease outbreaks may occur in aquaculture, and hopefully down the line lead to better ability to prevent the number of fish lost to disease and to decrease that number. And eventually, we'd also like this work to serve as preliminary data to seek additional funding with the aim of isolating Flavobacterium-specific bacteriophages that could be used either as preventative treatment or as reactive treatment to bacterial cold water disease outbreaks. And yeah, so if anybody would like to ask questions, I'd be happy to stick around a little extra to answer them, or I could go back to my email. So feel free to reach out to me that way as well, or drop them in the chat and I'd be happy to answer. So thank you very much for your time.
Great presentation, Brady. Yeah, you've done some awesome work and you've got some interesting stuff ahead of you.
Yeah, thank you.
Folks, if you've got a question, feel free to unmute yourself.
All right, I have a question.
Okay.
Yeah. Apart from carrying out molecular diagnostic tests and doing necropsy, did you carry histopathology to know histopathological lesions that could be observed in Great White Lake fish infected with Renibacterium?
Yes, so we collected samples for histology, but we haven't submitted them yet for analysis. But we do have those samples saved so that we're able to get those submitted for analysis at some point.
Sorry, which samples did you collect for histopathology?
The granulomatous lesions, we collected subsets of those into formalin. So yeah.
Okay. Thank you.
Yeah, absolutely.
And it looks like you have some interest in the chat too about the histological findings. So maybe you'll come back next year and give us an update.
Yeah, absolutely.
All right, well, let's call it there. Thanks a lot, Brady.